Good day, everyone. Our Module 3 is about biomolecules. I am Dr. Erwin Aberencili. For our learning outcomes, first, state the different elements and compounds present in a living system. Second, categorize biological molecules in terms of the increasing complexity of cellular organization. So let us begin with Lesson 1, Biomolecules and Overview. So in here, we are going to tackle in a glimpse the four biomolecules. So these are carbohydrates, proteins, lipids, and nucleic acids. So there are only 27 naturally occurring chemical elements that are indispensable in different living organisms. So the four most abundant elements in the Earth's crust are oxygen, silicon, aluminum, and iron, while the four most abundant elements in living organisms are hydrogen, nitrogen, carbon, and oxygen, which make up about 99% of the mass of most cells. In the illustration, as you can see, on the left side are the most abundant element in the body of an organism, particularly in a human organism. Oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus, wherein the number one is oxygen, which is 65%. And on the right side are other elements such as potassium, sulfur, sodium, chlorine, magnesium, and others. And this is around less than 1%. Meanwhile, water, which is a compound, a molecule, is around 62%, while protein is 16%, and 1% carbohydrates, 6% minerals, and 16% fat. So these are the properties of biomolecules. So biomolecules are organic molecules. So they are carbon-containing compounds. So not fundamentally different from other typical organic molecules. So meaning to say that biomolecules are somewhat the same as that of other organic molecules. So again, organic molecules are carbon-containing compounds. So they are the same types of molecules, react in the same ways, and obey the same physical laws. So in your in organic and organic chem, you're able to determine all these physical and chemical characteristics of organic molecules. So biomolecules behave the same way as that of the organic molecules. So on the right side are the four biomolecules that we are going to tackle. And as you can see here, that they are composed of they are composed of different organic molecules. So for example, carbohydrates, so it is composed of carbon, same thing as with protein, nucleic acids, and lipids. So some structure are in skeletal or line angle formula. Some are in using the expanded formula and so on. However, it doesn't matter, no. The point is that they are all carbon-containing compounds. So for the composition and structure, so biomolecules contain mainly carbon, which behaves as it always does in organic compounds. So basically, carbon has 4 valence electrons, so does it form 4 bonds, usually with a tetrahedral arrangement. So the carbon skeleton can be linear, straight, no branch. Branch meaning to say that it has a side chain. Cyclic, just like that of the cycloalkane. Or aromatic, meaning it has ring inside, or for example, benzene. So other important elements are H or hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So that's why if we are talking about the elements inside the human body, just think of John PS. Um, Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So again, I am showing the same picture from the previous slide because I want you to look into the different biomolecules wherein the carbon are those that are found on the corner. No, for example, in carbohydrates, this is actually a disaccharide, lactose. So those corner, except this one, this oxygen, and that one, no, these are all carbon. So carbon has 4 bonds. It can form 4 bonds. So for example, on this, just look at the arrow. So this is C bonded with to O, then bonded to H. And then this C is bonded to another C on this right side. And then this C, the same C or carbon, is bonded on the left side, carbon. So there are 4 bonds for carbon. So same thing, you can look on the different structure. For example, in protein, wherein there's double bond in oxygen. So same thing in here. So this is C bonded with C here, one bond, and bonded to nitrogen, and then double bonded with oxygen. So there are 4 bonds. And you can also check nucleic acids and as well as the lipids, but you will get the same 4 bonds because these are basic principles. So about 27 elements, as mentioned a while ago, are required by biological systems, including iodine and many metals, though most of these are needed in only trace amounts. So when we speak of that, it means that very minute or small amounts are needed in the system. So biomolecules contain the same types of functional groups as do organic molecules. So just think of the organic chem like that of the hydroxyl group, amino group, carbonyl group, carboxyl groups, and others. You may recall your organic chemistry topics about functional groups, where these are present in biomolecules. So again, I am showing the same picture, and let us determine some of this functional group. Okay, so for example, in here in protein, so we have here NH, no? So this is amino group. And then for nucleic acid, another NH. There's also OH group here, hydroxyl group in carbohydrates. In lipids, we have here carboxylic acid, so COOH. So same thing with other biomolecules. No, you can. These are all samples. No, examples. There are a lot of structures for each biomolecule. For example, in carbohydrates, no, there are monosaccharide, disaccharide, and polysaccharide, in which each has their own specific structures for carbohydrates. So same thing as with protein. So there are around 20 amino acids, and each amino acid has its own structure. Thus, you may look into the functional groups that are present among those amino acid. So another are the nucleic acids, no? So you can still check, for example, in adenine, in guanine, in thymine, cytosine, and uracil. And for lipids, there are a lot of examples that we can cite in lipids. And then you can still look into the functional groups that are present among the examples. But the point is that in biomolecules, you will see the same functional groups, same as that of the other organic molecules. So let us talk first about stereochemistry. So before we proceed to the specific biomolecule, so let us talk first about this one, because the structures that you will be encountering are in this particular subtopic, no, stereochem. This is very applicable in studying the biomolecules. So as in common with organic compounds, many biomolecules exhibit stereochemistry. So when four different types of atoms or functional groups are bonded to one carbon atom, the carbon is said to be stereogenic or chiral or asymmetric. If it is achiral, it is symmetric. But in this case, if there are four different types of atoms, just like in the example, that are bonded in the carbon, it is known as chiral molecule or asymmetric. So for example, in this one, carbon is found at the center. So there are four different types of atom. For example, we have here bromine atom bonded to carbon, chlorine atom bonded again to carbon, hydrogen atom, and chlorine atom, which are both bonded to carbon. So the carbon here is known as chiral center, and that the compound is said to be stereogenic. So as long as there are four different atoms, or it could also be four different functional groups, no? As long as they are different from each other, we can say that the carbon here at the center is stereogenic, and the compound can exist in two different isomeric forms that have different configuration and space. So in the different samples that you will encounter in biomolecules, you can actually identify carbon that are considered to be as chiral or not. So let us now move on to the to lesson 2, the four biomolecules. So I have here the illustration of lipids, nucleic acids, carbohydrates, and proteins, which are the four biomolecules that we are going to tackle. So let us talk first about carbohydrates. So the structures contain the same elements of C, H, and O. These are the structures of carbohydrates. So they are one of the macromolecules, so meaning to say that they are big molecules which contain carbon, hydrogen, and oxygen only with a ratio of 1:2:1. So the general formula for carbohydrates is that it is C or carbon, small letter N, H, 2, N, O, N. So for example, if you have 6 carbon, you'll have 12 hydrogen and 6 oxygen. So conserving the ratio of 1:2:1. So the C6H12O6 is actually the formula for monosaccharide sugar. So the term carbohydrates come from the Latin word saccharum, which means sugar. So additional information. So the carbohydrate that we eat, which is our main source of energy, is converted into glucose, which is readily used in the body. So it is classified as simple sugars or monosaccharide and disaccharide, and complex sugars as known as polysaccharide. So we are now going to tackle this classification of sugar, mono, di, and polysaccharides. So monosaccharides are sugar with one saccharide unit. So that means that it is a single unit of sugar. So monosaccharide is actually the building block of carbohydrates. So meaning to say that the carbohydrates are made up of different monosaccharide. So some of these examples are glucose, which is used as dextrose, and it is also the sugar found in the blood. So that's why whenever a person is sick and hospitalized, no, they usually put dextrose directly in the blood vessel, since dextrose contains glucose, which is the sugar found in blood. Another monosaccharide is galactose. Galactose is found in milk and other milk products. So actually, galactose is a product of the digestion or breakdown of um, disaccharide. And fructose, sugar found in fruits and honey, are monosaccharide. So they are actually isomer. So when we speak of isomer, they have the same molecular formula. The molecular formula of this monosaccharide sugar is C6H12O6. And then, but they differ on the structural formula. So just like this one, so you have here glucose, fructose, and galactose. Same chemical formula of C6H12O6, but they differ in the structural formula. So you can see they differ on the orientation of the OH and H. So for example, it is very obvious that fructose is different from glucose and galactose. But if you will look into the structure of glucose and galactose, they are almost the same, but still almost, only almost the same, but they are still different. Look at this one, no? The orientation of H and OH. Here it is found at the bottom. On this side, it is found on top. And this one is the same thing. It is found at the bottom, and this one is found on top. So that's one difference between the two. So the difference in their structures is the one that made also difference in their properties. So because though they have the same chemical formula, since they differ in the structural formula, so there is a difference in their boiling point. For example, boiling point of galactose has higher than that of glucose. Meanwhile, glucose is sweeter than galactose. But among the three, it is the fructose, the sugar found in fruits and honey, is the sweetest sugar. So let us now move on to disaccharides. So disaccharides are sugar with two saccharide unit joined by a glycosidic bond. So when we speak of glycosidic bond, it is the bond that attach one sugar unit into another. So meaning to say, it is that a bond, chemical bond, that bond the two monosaccharide. So for example, we have maltose is a disaccharide formed from two glucose units and is found in malt. So meanwhile, sucrose found in regular table sugar contains glucose and fructose units. So lactose found in milk and milk products is a disaccharide containing glucose and galactose unit. So if you'll notice, in all breakdown of disaccharide unit, there's always glucose. There's glucose in maltose, there is glucose in sucrose, and there is glucose in the breakdown of lactose. So there's always glucose because this is needed again by our blood. So this is the structures of the disaccharide sugar. So the first one is the sucrose. On the left side is the glucose, and then the right side is fructose. So this one at the center is known as the glycosidic bond. On the other hand, we have here lactose. Again, there's glucose, then there is galactose on the left side and glucose on the right side. And these are actually again glycosidic bond. And then for maltose, there are two glucose units. So meaning to say, when you digest maltose, you will always have two glucose units. So that means there will be an increase in the blood sugar. By the way, maltose can be found in beer. No, so those people who love or habitually drink beer have lots of sugar, so that's lots of carbs, leading to a beer belly. No, nakakalaki ng tiyan because of the carbs. Now you know, right? All right, so let us now proceed to polysaccharide sugar. So it contains a lot of saccharide units, around 250 to 400 glucose molecules that are connected via alpha 1,4 glycosidic bond. So that alpha 1,4 actually is the number of carbon, meaning to say that the first carbon is attached on the fourth carbon of the next molecule. And then the other side is another 1 and 4, then 1 and 4, and 1 and 4, and 1 and 4, and so on and so forth. So the starch and cellulose are polysaccharides. So another is glycogen. So I have here the illustration. We cannot put the entire structure because, again, as mentioned, there are around 250 to 400 glucose molecules that are connected by a glycosidic bond. Now, on this example, we have here starch. You'll notice that all the CH2OH are found on top. So the glycosidic bond, this one, the one that connects the two glucose, there you go, and then repeat, meaning to say that it is a chain. No, it is a polymer, polysaccharide. It is a continuous chain of around 254 to 400. And then, by the way, starch is found among, it is a storage form of glucose in plants, just like those that are, just like sweet potato, and other things, other starch. And then cellulose, on the other hand, is found in the cell wall of plants. So the structure looks like this one. So this is composed again of around 250 to 400. And then glycogen. Glycogen is a polysaccharide sugar found in the liver of mammals, or any other organisms with liver. So glycogen, when there is oversupply of glucose in your blood, so the insulin hormone will give signal to the liver to absorb the excess glucose. However, the liver cannot absorb and store. No, they cannot store glucose. It must be converted first to glycogen so that it can be stored. So that's why the glucose from the blood in excess are stored as glycogen. And then when the blood needs it again, the liver will release the glycogen in a form of glucose again in the blood to be used by the cell to form ATP, or adenosine triphosphate. And then let us now move to proteins. Okay, so protein was believed to be the beginning of life. So that's why they thought before that proteins are actually nucleic acid. So it comes from the Greek word proteos, which means first rank of importance. Carbon, hydrogen, oxygen, and nitrogen are the elements that make up a protein. So sulfur and other metals can also be found in some proteins, because sulfur and other metals are actually found in some amino acids. So proteins are one of the macromolecules that are made from multiple units of simple molecules called amino acids. So the building block of proteins are the amino acids. So there are around 20 amino acids, 10 of which are considered to be as essential amino acid, and the other 10 are known to be as non-essential amino acid. So when we speak of essential, these are needed by the body, and it must come from our diet. It cannot be, these are not manufactured by our body, unlike in non-essential amino acid, wherein the non-essential amino acid are manufactured by our body, but and we do not get it from our diet. Though some can be taken from the diet, but since it is being manufactured, no, by our body, so it is considered to be as non-essential. So the different amino acids, these 20, are connected to each other to form proteins, and the chemical bond that attach an amino acid to another is known as polypeptide bond. So this is the general structure of amino acid. So if you'll notice here, there are two functional group. No, we have here the carboxyl group, COOH, or the carboxylic acid side, and this is the amino group, or the NH. And of course, you have there the side chain. The side chain could be an alkane or anything that is considered to be as a side chain. So these are the examples of proteins. First, we have collagen. Collagen found in connective tissues such as tendons, ligaments, skin, cartilage, and the cornea of the eye. So this is an example. This is the illustration of collagen. You have here collagen fibers, collagen fibrils, then collagen molecules, and then you have here the amino acid chain. So this is alpha chain, and do not worry about some other terms, because this is just a glimpse of the biomolecules. Of course, when we go to these topics, we will discuss the detail of it. So we have here the alpha chain, and then the amino acid, and then the illustration just zoom it out or zoom it in so that you can be able to see the structures of the amino acid chain. So that's why here in the bottom side, you have here the different names of the amino acid. So glycine, proline, glycine, proline, glycine, proline. And then, and these are found inside the collagen in a form of protein. So meaning to say that if you are going to dismantle the collagen fibers, what you can get are actually proteins in the form of amino acid. So another one is fibrin or silk protein. So one of the strongest natural fibers found in silk. Now, silk are actually produced by the silkworm, and some are produced by spiders. So for example, in this one is the spider silk. So different types of silk serve diverse biological needs. And then this is the fiber from the spider silk. And then this fibril are well-aligned beta sheets cross-link and reinforce polymer network through strong intra and intermolecular interactions. So these terms meaning to say that the proteins are bonded together in a beta sheets. No, so there are different forms of protein, and one of them is the formation of the alpha and beta sheets. So this one is beta sheets. And then you have here beta sheets, B turns, ensure strength. So if you are going to zoom it in a polypeptide backbone, you have here the amino acid that are linked together by polypeptide bond as well as hydrogen bonds. So that's why silk protein is one of the strongest natural fibers found in silk. Okay, or fibroin. Okay, so another example of protein is the keratin, a fibrous protein in hair, skin, and nail. So in your anatomy, for sure, this was discussed, but not in a chemical form. So this is the chemical structure of the keratin. Okay, so you have here on the left side is the ball and stick model of the keratin as a protein, but on the right side is it contains the formula, the chemical formula for the keratin. So each one, again, are the amino acids that are linked together by disulfide bonds. So disulfide bonds meaning to say that sulfur to sulfur, and then hydrogen bonds, H to H. Okay, and of course, there's another, the polypeptide or the peptide bond. So again, keratin also is a strong protein because of too much bonds among the elements that are found in the formula. Another is myoglobin. No, contains a heme group. No, contains an iron 2, ion as its center where oxygen is being stored. So if you are going to look at the structure of the myoglobin, so you have here the myoglobin in 3D. In ball and stick model, we have here the myoglobin showing the structure of the protein. So there are actually 4 structures of the protein. And then here is on the right side is a zoom chemical structure of the heme or hem, showing the iron at the center of it. So you have also here the methyl group and the vinyl group and the propionate group. So which are all organic compound functional groups. And then in connection to that, you have here hemoglobin. It is a globular protein that carries oxygen from the lungs to the bloodstream. Now, hemoglobin, if you'll notice, it has the heme structure. That's why hemoglobin has iron on it, no? So the one that gives also color to the blood when encountered by oxygen. So hemoglobin is a globular protein that carries oxygen from the lungs to the bloodstream. So of course, this is a definition for the physiological purposes, but in here is the chemical structure of the hemoglobin. So this one has the alpha chain 1, and then the beta chain 1, beta chain 2, and then the alpha chain 2, and then the heme structure that has iron 2 on it. So making it possible for the blood to carry oxygen because of the heme structure. So another one, which is, again, this is the last part of the examples of protein, are the enzymes. So enzymes are proteins, no? And they catalyze chemical reaction. So they speed up chemical reaction, but they themselves are not part of the reactants nor the product of the chemical structure. So these are some examples of enzymes with specific function, lipase, pepsin, and sucrase. So lipase help in the digestion of fats. Pepsin help in breaking down of proteins into peptides, and sucrase help in the digestion of sugars and starches. So you have here the role of the lipase that helps in the digestion of fats. So you have here triacylglycerol with water. So with the help of the lipase, triacylglycerol are broken down into the product. No, these are the glycerol and the fatty acid. And then we have here pepsin. So this is the hydrolysis of peptide bond. Of course, though there is a hydrolysis or the addition of water, there is a role of pepsin that helps in the breaking down of protein. So this one on top is the protein into smaller unit of peptide and then amino acid group. And then this one, the sucrase that helps in the digestion of sugars and starches. We have here on top is sucrose. Sucrose is a disaccharide sugar, and with the help of the sucrase, the enzyme, and then of course water. So this is separated in or broken down into two monosaccharide unit, which are glucose and fructose. And then let us now move on to the third biomolecule. We have here lipids. So lipid come from the Greek word lipos, meaning fat. So that's why the term is liposuction, to suck in the fat in the body of human. So lipos. So from lipos is the word lipids. So lipids are a family of biomolecules grouped together because of its property of being hydrophobic or water-bearing. And lipids are also non-polar molecules, making it soluble to non-polar solvents like acetone, ether, and benzene. However, lipids are insoluble in water. So this is the classification of lipids. So you have here lipids, and then lipids are classified as fatty acid and then steroid at the bottom. So fatty acids are classified into prostaglandins, and then it is also classified as waxes, triacylglycerol, and glycerophospholipids, and as well as sphingolipids and glycosphingolipids. Meanwhile, lipids that are classified as steroids are further classified into cholesterol, bile salts, and steroid hormones. On the other hand, the structure of the classification of fatty acids include four waxes. You have here fatty acid and then a long chain of alcohol. And then for triacylglycerol, you have here the glycerol that are bonded into three fatty acid. And for glycerophospholipid, you have here glycerol, two fatty acid, and then phosphate group, PO4, and then amino alcohol that is attached to the phosphate group. And then for sphingolipids, you have here sphingosine that wherein it is attached to one fatty acid and then to one phosphate group and amino alcohol. And then for glycosphingolipids, you have here sphingosine attached by a fatty acid and then sugar. So that is the classification of lipids. But then again, we are going to discuss further on that when we go to the specific topic about lipids. So fatty acid is a chain-like molecule containing a long chain of carboxylic acid. And fatty acid can be saturated. When we speak of saturated, it contains only single bond in its long chain hydrocarbon chain, or unsaturated contains double bond. So when it comes with which one is healthy, unsaturated fatty acids are actually healthier than that of saturated fatty acid. Actually, most saturated acid can cause the different heart diseases because of the structure having only single bond, unlike in unsaturated fatty acid. So since lipids are nonpolar molecule, it is insoluble in water. So that's why if you are going to wash your kitchenware, which are uh, with fatty acid or with fats on it, so you cannot remove the fats with just water. So you have to use soap, no, this dishwashing liquid or dishwashing soap for you to be able to remove the fats on the kitchenware, since they are considered to be as nonpolar molecule. So saturated fatty acid is a straight chain where all carbon atoms in the chain have two hydrogens singly bonded to making the molecules to form a strong attraction. And then because of this strong attraction, saturated fatty acid have high melting point and are solid at room temperature. So unsaturated fatty acid, on the other hand, tends to bend due to the presence of double bond in one of the carbon in the chain. So that's why in most commercials of food featuring fatty acid, so they are using the term unsaturated fatty acid since these are actually healthy. So the molecule will not be as close as that of the saturated fatty acid. Unsaturated fatty acids have a low melting points than saturated fatty acids. So here is the general formula for the fatty acid. You have there the head and the tail. So the head is the red one, so it is composed of carboxyl group. And then the tail is composed of hydrocarbon chain. So in the saturated fatty acid, so we just reverse the structure. So the head is the COOH here. And then this one, no, this lauric acid, long hydrocarbon chain. On the other hand, you have here unsaturated fatty acid. So in the form of palmitoleic acid. So it has one double bond found on the 7th and 8th carbon. So you have here 16 carbon, including the C for the COOH. So what makes it healthy or unhealthy in case of the saturated? Since the saturated fatty acids contain all single bond, so they stack well together. So when there is a low temperature, so they stack well together. Though sometimes it doesn't need to have a very low temperature. So sometimes when you leave the saturated fatty acids unattended and you do not put it in any other place, you'll notice that the saturated fatty acids gradually or slowly turn into solid. On the other hand, the unsaturated fatty acid containing a double bond tend to bend, no? So once they bend, and you put them all together in one place, so they do not stuck well together because of um, the structure is not evenly and it is not uniform. So thus, it is liquid in room temperature. So it is healthy actually. We have another triglyceride. So are lipids that contain glycerol backbone and three fatty acid. The three fatty acids connected to the glycerol backbone are not necessarily of the same kind. So fats and oil are examples of triglycerides. Fats are usually from animal sources, contain mostly saturated fatty acid, making it solid at room temperature. So versus oil, oil are liquid and it is found in among plant sources. So it contains unsaturated fatty acid and it is liquid at room temperature. So that's why if you're going to use any of this, you make sure to use oil that are coming from plant sources since these contain unsaturated fatty acid. So this is the structure of the triglyceride. So it has a glycerol that are bonded with a three fatty acid chain. So let us now move on to phospholipids. So it is another type of lipids that contains glycerol, two fatty acid, and a phosphate group. So phospholipids, unlike other kinds of lipids, it has a polar end. When we say a polar end, it is the hydrophilic end. The hydrophilic end is a water-loving, meaning to say it attracts water, which is the phosphate group, and nonpolar end or the hydrophobic or the water-bearing, the fatty acid group. So the dual property of liquid allows the phospholipid to form a bilayer. So actually the phospholipids are found in the cell membrane of the cell. So that's why it is known to be as bilayer because of the presence of phospholipid. So that's why in the definition or description of cell membrane, there's always the term, it is a phospholipid bilayer. So that's the structure of the phospholipid. So you have here on top the hydrophilic or the water-loving head, and then the hydrophobic tail on the bottom side. So it contains phosphate group, this one, glycerol, this one, and then the saturated fatty acid, and then the unsaturated fatty acid. So that depends on whether these are both unsaturated or these are both saturated fatty acid. So let us now move on to the last biomolecule. So we have here nucleic acid is most important macromolecules that serve as a storage of genetic information of the cell. So it is very important simply because all the information necessary for the life of the organisms are stored in as the genetic information of the cell. So it is made up of nucleotide monomers that contain a nitrogenous base, a pentose sugar, a phosphate group. So there are two types of DNA, of nucleic acid. These are the DNA and the RNA. So the DNA or the deoxyribonucleic acid is the blueprint of life, carries the genetic material, and provides information for its own replication. So DNA is found in the nucleus of the eukaryotic cell. So it is a double helix structure composed of a sugar and phosphate backbone and complementary bases of adenine, thymine, cytosine, and guanine pair. So that is purine to purine to pyrimidine, pyrimidine to purine. So we're going to tackle that later on. So for the RNA, it carries information from the DNA for the protein synthesis. So it has three types of RNA. These are the mRNA or the messenger RNA, rRNA or the ribosomal RNA, and the transfer RNA or the tRNA. So this is the structure of the nucleic acid that has carbon, carbon-containing compounds, and these contain also the functional groups. So you have here the phosphate group, the PO4, this one. And then you also have here that is bonded with a deoxyribose, the sugar. So this is the backbone, deoxyribose. until the end. And then you also have here the purine bases and the pyrimidine base. So for the nucleic acid or the DNA to be uniform in size, so they found out that the partnership among the pyrimidine bases should be should be purine to pyrimidine or pyrimidine to purine. So in the RNA, this is the ribose. So instead of deoxyribose, it is composed of ribose. And then uracil also replace thymine as a pyrimidine base. So that's why that's one of the difference between the RNA and the DNA. All right. So do you have any questions regarding this topic? So if you do not forget to bring that up during our discussion, or you may send me a chat or an email regarding your questions and clarifications about this topic. So thank you so much and see you on our lecture next week.